Hi everyone, welcome to this tutorial, and in this tutorial we will be looking at the life cycle of malaria parasite, and taking you through this tutorial it's me, the physio tutor. So before we can dive into this tutorial, I want you to understand that as we'll be talking about malaria, um, there are several conservative agents of malaria, of which we have more than 125 species of the Plasmodium which are in existence. So, but human infection is actually only carried out by five five species, which are the ones which um, bring about human infection of malaria. So we have the the Plasmodium vivax. We also have the Plasmodium falciparum. We also have Plasmodium malariae. We have Plasmodium ovale, and we also have Plasmodium knowlesi. So these are the five major Plasmodium species which are linked to human infection. Okay, so but in this tutorial we are just going to consider the life cycle of the malaria parasite. So talking about this very malaria parasite, in this cycle, when we're talking about the host, there will be need for two hosts are needed in this cycle. We have the definitive host, and we also have the intermediate host, of which we already discussed about these things. If you didn't watch our previous video on the introduction to medical parasitology, please make sure to watch that video. Very important. So when it comes to the hosts, we have the definitive host, and this definitive host basically happens to be the female Anopheles mosquito, where sexual cycle basically takes place. And then we also have the human, which tends to act as an intermediate host. That is where we have a sexual cycle taking place. So now let's get into this and understand exactly what happens in this cycle. Okay, so this slide basically shows us the whole cycle the uh, of the malaria parasite. So we have the whole cycle right here. Okay, so the malaria cycle, as you can see from this slide, it basically contains several key stages. Okay, it has several key stages, of which the first one we are going to categorize it as transmission. Okay, so the first stage we are going to consider it to be transmission. As you can see right here, this slide says if this man gets infected by the bite of infected female Anopheles mosquito, which contains sporozoites in salivary glands. Okay, so we have transmission here. So we have transmission here where that is from mosquito to human. Okay, so man here, as we can see, gets infection by the bite of infected female Anopheles mosquito, which contains sporozoites in salivary glands. Okay, so these sporozoites basically are the infective form of the parasite. So the sporozoites are the infective form of the parasite. Okay, then from there, on stage two, or on step two, we have what we can refer to as the liver stage. Okay, we have what we can refer to as the liver stage. So this liver stage is also referred to as exoerythrocytic stage, which is also known as the pre-erythrocytic schizogony or intrahepatic or tissue stage. Okay, so what happens is that these sporozoites in stage 2, the sporozoites tend to travel to the liver, as you can see. Okay, so the sporozoites tend to travel to the liver and infect the hepatocytes or the liver cells. Okay, so you'll find that once these sporozoites have entered into the liver, inside the liver cells, they mature and multiply into um, merozoites. Okay, so they mature and multiply into merozoites. But now, as you can see right here, there are these species, because these are sporozoites. So some, they just go straight there and mature and form merozoites. But there are others, okay, some species such as the P. vivax and the P. ovale. So the P. vivax and the P. ovale can actually form what we call hypnozoites. Okay, they can form hypnozoites which will remain dormant. Okay, so when these hypnozoites remain dormant, are the ones which can cause relapse later. Okay, they'll bring about relapse later. So talking about relapse, it is very, very important that we get to differentiate relapse from another phenomenon that is usually seen in Plasmodium falciparum and also Plasmodium malariae, which is what we call recrudescence. Okay, well, what we call recrudescence. So there is a very uh, slight difference, yet significant, between relapse and recrudescence. Okay, so when talking about relapse, relapse basically occurs due to the reactivation of dormant liver stage parasites, which we call hypnozoites. Okay, so you'll find that after the initial infection, these hypnozoites, which were dormant right here in the liver, they can they can remain inactive in the hepatocytes for weeks, months, or even years before reactivating and causing a recurrence of malaria symptoms. But now, when you're talking about recrudescence, this just refers to the recurrence of malaria symptoms due to the survival of blood stage parasites that were not completely eliminated by treatment. So it's very, very important that you get to understand that difference. Okay, so that was the stage 2 um, of this cycle, which is the exoerythrocytic stage. Now, on stage number 3, so we had stage 1 here. Okay, so we had the stage 1 right here, and then we had our stage 2, which is the liver stage. Then uh, we are going to have our stage 3 right here. Okay, so this will be our stage 3. So in this, on stage 3, this is what we are going to refer to as the blood stage or the erythrocytic stage. Okay, so it is this very stage, which is the blood stage, that brings about the manifestation of symptoms. Okay, so what happens? Remember, the the um, the sporozoites matured in the liver, and when they matured, they formed the hepatic merozoites. Okay, they formed hepatic merozoites. So as they formed those hepatic merozoites, you'll find that the hepatic merozoites will now be released into the bloodstream. And when they are released into the bloodstream, what actually will happen is that, as you can see here, they are being released into the bloodstream. And when they have been released into the bloodstream, this arrow is showing you that they will now invade the red blood cells. Okay, so you'll find that these merozoites, actually, they are going to bind um, to certain receptors. Okay, they are going to bind certain receptors on the red blood cells' surface. And when they do bind, what happens is that they will enter the red blood cells by a process known as endocytosis. Okay, so you'll find that the entry process into the red blood cells actually takes approximately about 30 seconds. Okay, so it is very, very important that we make mention that when the Plasmodium species is actually transmitted by blood transfusion or placenta, the merozoites are the ones which are going to act as the infective form of this parasite. Okay, so now what happens is that, as you can see here, on the same stage 3, what will happen is that inside the red blood cells, the merozoites will actually develop into what we call trophozoites. Okay, merozoites will develop into what we call trophozoites. Okay, so this trophozoite is actually considered to be the feeding stage of the parasite. Okay, feeding stage of the parasite, in which later now it on undergoes several nuclear divisions, and when it undergoes several nuclear divisions, it then matures into schizoids. Okay, schizoids, and then those schizoids will they when it matures into schizoids, that is from the trophozoites, then they we are going to have the schizoids. Okay, so this is the early trophozoite on the ring form, and then we have the late trophozoite right here. Okay, so you need to understand that the plasmodium actually feeds on hemoglobin. Okay, the plasmodium feeds on hemoglobin. So the during the schizoid stage, the parasite will then divide, and when it divides, it produces approximately 8 to 32 new merozoites, of which later there will be the rupturing of the red blood cells and the release of um, erythrocytic merozoites. Okay, so when those erythrocytic merozoites, when they are released, these are the ones right here on this part. Erythrocytic merozoites, when they are released, they are being released for them to infect more red blood cells. Okay, so it's very important that you note that early trophozoite form is considered or is called the ring form. Okay, very, very important to take note of that. Okay, so it is very important that you understand that on stage number 3, which is the blood stage, at the end of the blood stage, red blood cells will then rupture to release the daughter merozoites. Okay, release the daughter merozoites, malarial pigments, and toxins into the circulation, which will result into malarial um, uh, paroxysm of fever at the end of each erythrocytic cycle. Okay, now since we've mentioned about fever, okay, we've mentioned about fever, it is very important that we bring in a concept that we call incubation period. Okay, incubation period. So talking about the incubation period, this just refers to the time interval between the entry of the parasite to the body and the appearance of the first clinical feature. That is the incubation period. Okay. Now um, the trophozoite stage is actually also considered to be the diagnostic stage of malaria using a microscope. Okay, so that was on stage number 3, where we have the release of erythrocytic merozoites. Okay, then we also have the last, the fourth stage. Okay, we have the fourth stage. So on the fourth stage, this is what we call gametogony. Okay, we have what we call gametogony. So what happens is that there are some merozoites which will actually transform into gametocytes. Okay, there are some merozoites which will actually transform into gametocytes. Okay, then now you need to understand that the gametocytic development actually takes place in the blood vessels of internal organs, for instance the spleen and the bone marrow. Very, very important to actually take note of that. Okay, so this is the arrow here on step 4 where we are. So we have two types of gametocytes which are in existence. We have the male gametocytes, which are also known as microgametocytes. Then we also have the female gametocytes, which are also considered to be macrogametocytes. Okay, so Talking um, about stage number 4, that is what happens. Okay, so some of the merozoites will form gametocytes. Okay, then these gametocytes will be transmitted to the mosquito. Okay, so when we reach there, then you'll find that we have the cycle like that. So, in closing with the life cycle of the malaria parasite, you need to understand that individuals harboring gametocytes are considered as carriers or reservoirs of infection. Okay, so a patient can actually be a carrier of several Plasmodium species at the same time. However, it is very important to mention that gametocytes are effective in transmission of the infection if they are, number one, mature enough, number two, they are viable, that is, they are alive, not dead. Then, lastly, they uh, they are present in sufficient density, that is 12 uh, cubic millimeter, okay, 12 per cubic millimeter of blood. Very, very important to take note of that. Thank you very much. That is where we end this tutorial.